This is Audiommunity, a podcast about our body's never-ending fight with the outside world. Hi everyone, this is Audiommunity, recorded May 3rd, 2016. I'm Kevin Bonham, and I'm joined today by Matt Woodruff.
How's it going, everyone?
And no Kate today. She is apparently slammed with stuff. I think maybe the union busters got to her, carted her off, and have her in a dang cell somewhere.
Be. I think I heard. Does she have high school students? Is that something that she does? She said
Not
she had a
student
or something along those lines, or
maybe an undergrad. I don't know. Maybe.
Anyway.
It sounds like she's doing way too much. She should be graduating any day now.
Right.
But she decided that she would also start a grad student union, and also she's trying to figure out what she's doing with her life.
Right.
And also mentoring students, maybe. So,
I mean, kudos to her for blowing it up as she goes, right? I feel like that's really what you need, is someone that is close to the finish line and then looks back and says, this process isn't what I hoped it would be, and then just sort of like explodes everything on the way out. So hopefully that will help other people. We'll see.
Yeah. That is using some really awesome technology that was originally actually developed for neuroscience today. And some enterprising immunologists decided to turn it to the problem of B-cell germinal centers. But before we do that, Matt, what are you drinking this evening?
Yeah, so I was drinking a Sierra Nevada Nooner, which is their Pilsner.
I have not had that one.
Yeah, so, I mean, I think you and I are both sort of on the same page with Sierra Nevada in that I've never had a bad one. And so anytime I see one that I haven't had, I have a tendency to buy it. So, I have to say...
That seems like a good policy.
Yeah, it is. It is, I think, generally. It's okay. It's
that's interesting.
Not fantastic, but it is definitely a Pilsner. I think it's definitely headed towards the kind of macro brew style. So, it's not the really crisp German Pilsners that I tend to get into, but, still pretty good. And unfortunately, I'm not drinking it now because I was relegated to the bedroom today. So I'm currently sitting on a bed with a lap desk. My microphone is on the lap desk, and it didn't seem like it was appropriate to be drinking beer in bed. So I drank the rest of it. So we'll see when that kicks in.
That makes sense.
Right.
So we should say that any audio problems you notice from Matt, like some bumping, random scraping, I may just leave it in instead of trying to edit it out. But you should know that that's all Matt.
Right.
Yeah.
Kevin has now been given license to throw a party in his end of the world.
Exactly.
All be on me.
If I had anything other than warm beer to throw a party with. I'm drinking the Grey Lady that I purchased for our last recording, which was now like 4 weeks ago.
Yeah. Just
as an aside, every single time we record a podcast, I say, this time I'm really going to get better about recording or about editing. And I do have 2 episodes still in the dock ready to go that we recorded months ago. But I found out recently a way to basically double the speed at which I can edit these podcasts. So for really reals this time, I think we're going to improve the speed.
You know what I think
editing.
it's like? It's sort of like, so we have a lot of bioinformaticians in the lab, and I get the sense that with bioinformatics problems, and you know this as well as I, there are 2 things that you can try to do. You can either try to get an answer out of the data, or you can try to build software that allows you to get the answer out of the data better and faster. And I feel like we sort of go back and forth between, oh my God, we just need to get this podcast edited and out. And then sometimes we make leaps and bounds as far as I just realized that I was wasting 14 hours editing this last podcast. So
yeah, no, that's certainly true for bioinformatics. That's what I've been doing for the last 2 years. And it's really sort of amazing how you can, this actually happened to me fairly recently. We're trying to write up a paper on the microbiology work that I've been doing recently. And it was like, I spent literally 24 hours. I mean, 3 solid 8-hour days at the, in my text editor, trying to write Python code to parse this one column of data and figure out precisely what the order of things, it doesn't matter what it is. But basically, after I showed this to my PI and I was like, okay, here's what I've accomplished. And it's nearly there. We're almost there. She was like, can't you just like click the sort button on Google
Oh
Sheets for
no.
this column.
Oh
And I was like,
no.
no, of course you. Oh, oh yeah, yeah, you can do that, can't you? And it was just like,
Well,
it's sometimes, most of the time that doesn't happen. Most of the time it actually takes an algorithm, but sometimes, yeah. So yeah, so basically I figured out how to listen to our podcast at double speed in my editor,
there you go.
so that as I
Perfect.
scan through looking for places where we're burping and saying curse words that I have to fuzz out, I can do that now twice as fast, basically.
Nice.
Or three
Nice.
times as fast, although then it gets kind of unintelligible.
Yeah. Although, weren't you listening to audiobooks at like 4 times speed at one point?
Oh, yeah, no, I mean, three times.
Three times.
But when you have audiobook narrators, typically they have wonderful enunciation and are speaking in very, very slow speech that is quite understandable. And so if you speed it up really fast, then you can sort of still understand it.
Got it.
Yeah, so, but 2x speed, I can totally follow I listen to most of my podcasts at 2x speed anyway, so while I'm editing, it sounds normal.
Well,
So
there you
that's
go.
good.
We'll see how that turns out.
Yeah. So in principle, guys, this is May 3rd. In principle, this podcast should be posted by May 10th. So hold me to that.
I'll try.
If it's not May 10th, then I'm just going to edit out this section and then nobody will ever know.
I will post it. I will post the raw.
Okay.
It's sort of like Big Brother with the big editing rooms. You just go back and redact all of the things that aren't supposed to have happened.
Mm-hmm. Yeah.
Anyway,
Absolutely.
so speaking of Big Brother, science.
Right.
Yep. So I'm going to go ahead and admit that there was a selfish component. I think we mentioned in a previous recording how these papers get tossed around and how we decide to select papers to talk about on the podcast. And sometimes it's just like random things. I think one of the podcasts that we're going to be editing is the Kiwi podcast. Actually, is that already out?
No, that one's already up. Yep.
Yeah, so
that one went up a couple days ago.
You know, that was selected largely because it had the words kiwi and allergy in it, and that seemed like a cool thing. There was some transplantation stuff that was weird. This one was a little bit different. This one I selected because I just worked on an F-32. So this is a fellowship through the NIH on the project that I'm currently working on. And so I think I've spoken a little bit about the vaccine work that I'm doing in Bali Palundran's lab. But basically, as I was writing that fellowship, this paper, along with another paper by Garnet Kelso's group, came out basically back to back. And it totally changed the way that the introduction to my fellowship needed to be written. So there's a lot of historical understanding, and I would say that this is sort of typical of the vaccine field. I think that there's a lot of dogmatic understandings around some really general concepts in vaccinology, and then every once in a while something will come out, and people look around and be like, wait, we didn't know this before? How in the last 30, 40 years that we've been looking at this, didn't we, really have this basic conceptual understanding? And so, yeah, so these two papers essentially formed almost the entire introduction of my fellowship. And so I was...
And I want to interrupt briefly and say, like, a lot of times people, like, when you say that two papers changed the way that your intro had to be written, like, most of the time when we write an introduction, we sort of, like, know what we're going to say. We write it, and then we go search for papers that actually, demonstrate the things that we're saying, because we sort of already know, and it's sort of just... so that's pretty incredible. Like, people have this idea that often in science we have these sort of revolutionary things that change the understanding, and most of the time that doesn't actually happen. Most of the time, science is just sort of moving along at a steady pace.
Right.
So that's a important thing to say.
Yeah. And so it's, it's not like it invalidated anything that had come before it, but it was definitely a huge addition into the way that we need to understand how vaccine responses actually work. And on top of that, I mean, this paper's sort of in my wheelhouse, to be honest with you. I did a lot of imaging work during my thesis, and this takes advantage of some really, really cool imaging technology, as well as some next generation sequencing technology. I mean, this is like the prototypical science paper for me. It has a huge impact on the field. It's got really sexy technology. They definitively show the things that they show, and it's like 4 figures long.
And
So,
super pretty pictures.
oh, and it's gorgeous. It's absolutely gorgeous.
We're not going to make a comic for this episode. I'm just going to cut out one of these fluorescence images and post that because it's really beautiful.
Yeah, and so we should say that in case people are out at academic centers where people come and give talks, the last author on this, Gabriel Victora, who was a former postdoc out of the Nussensweig lab. He was at Rockefeller for a long time, but he's now at MIT. But if he comes to give a talk, definitely go, because this will be a sexy presentation to watch. So
and let's just say that the first author there is Jerome Tass.
Right.
And there's a bunch of other collaborators as well, but we'll post a link in the show notes.
Yep.
To the paper.
So anyway, yeah, I feel like we can just sort of jump right in on the science then. So I mentioned before that there's a lot of dogma surrounding vaccine biology, and one of the reasons for that is that obviously we had limited tools in how we looked at vaccine biology leading up to the 1990s, early 2000s. There was just a limitation in the sorts of readouts that you can get. So you can vaccinate with something and you can get a pretty good antibody response, and you can measure those antibody responses, but it's really hard to tell what that antibody actually recognizes, right? So we know we've talked about B cell receptors in the past. So B cells are going to be your antibody producing cells. And so they all individually have this unique B cell receptor that they carry around with them. And the shape and charge of that B cell receptor tells the B cell essentially what it's specific for. So let's say you have a B cell and it's got a certain receptor, and HIV walks along and HIV binds to this receptor really tightly. The B cell wakes up and says, oh God, I found it. I found something that I'm specific for, right? And so through a series of steps, we talk about T cell help in the past. B cells can't just become activated well by themselves. They require some help. But over that process, B cells will differentiate into plasma cells, and those plasma cells will release antibody against whatever it was that B cell was specific for.
And can I interrupt here for just a sec? Because I want to mention a misconception that I've realized that some of my students have here. So when we talk about B cells having these different receptors, and we've talked before about VDJ recombination and all of that stuff, what we're talking about here is like maybe 5%, probably less than that, 1% of the total protein is actually different between different B cells. The vast majority of that B cell receptor or antibody gene, or rather the protein that is coded for by that gene, the vast, vast majority of that is identical between all B cells. Right? And so even though we have this chunk that we call the like variable domain or the antigen binding domain of the antibody, and that's takes up a big chunk, the actual thing that is variable is this tiny, tiny piece. So that goes really into why you're talking about it's hard to find what that is amongst all of your different B cells, because most of it is identical.
Right. And so, so this gets into sort of the nature of a B cell, of a humoral response, right? So each individual B cell, like you said, they're pretty close to identical to each other, except for, and really even within the variable region, it's just these complementarity determining loops, right? So these are very, very small regions of the antibodies that are actually different, and they tend to be a little on the flexible side. They don't have a ton of structure to them. It can be very difficult to know what the complement determining loops. I'm just going to call them CDRs because I'm going to go back and forth there. So it's very difficult to determine what the actual structure of those CDRs look like. And so if you vaccinate a random mouse, so let's take a Black 6 mouse, and I'll stick with the model system that I use actually in the lab because I know just a little bit more about it, but I vaccinate with rabbit serum albumin. And so every mammal has some sort of blood albumin that floats around in it, right? And this makes sure that hydrostatic pressure essentially is in equilibrium in your blood. It's a maintenance protein that floats around. So everybody's got one, but if you take this protein and it's really abundant in the blood and you take it out of rabbits and you vaccinate it into mice, then you get this really robust antibody response against that albumin protein, right? But when you're looking at that antibody response, people have a tendency to believe that antibody response is monoclonal, right? I think that it is just a general way that people think about it. So you expect, oh yeah, there was an RSA, rapid serum albumin, RSA specific B cell, and it saw this albumin, and you create this response, and now here we are. All of these antibodies are sort of coming from this original precursor. And anybody that thinks about vaccine biology hard, I don't think comes up with that in their head, but I think that that's generally, when you're talking even to immunologists, that's sort of the model that everyone goes by. But realistically, there is this huge competition that's going on. So when rabbit serum albumin finds its way into a lymph node, it's not just one or two B cells that are going to be specific and are going to see the outside of this thing. Presumably, you're going to have tens or hundreds of precursor B cells that are all going to be able to see some part of the outside of this protein, right? And that's really important because, as it turns out, in something like HIV, so I've mentioned HIV a couple times, HIV has this protein on its exterior, GP120, and it's the protein that HIV uses in order to attach to human cells. And for decades now, we've known that. And for decades, we've been trying to vaccinate people against GP120. And by and large, we're really good at it in that we can get really good antibody responses against GP120 in humans. The problem is that they're...
so we've cured HIV.
Oh, yeah.
Sweet.
Yeah. Over and over again. No, but they're totally useless, right? So you can get good antibody responses out of humans against the correct proteins, but it turns out that that's not the important part. The important part is that there are these critical residues within the HIV protein that serve as targets. And if you don't hit those residues, then you're not going to inactivate the protein and you're not going to prevent HIV attachment. And so this is a major issue in the field where you have all of these B cells that all recognize the same protein. Not all of them are going to form a fully functional immune response, which means there's some sort of restriction that's happening in the process. And we know that restriction to be the germinal center, which is what this paper is going to center around. But during the restriction process, you're actually going to lose a lot of the targeting of a lot of the different sites around the protein. So what you end up with is a diverse antibody response, but that diversity is restricted from the initial subset of B cells that could have potentially responded, right? And it used to be that when people talked about vaccine biology, they were looking at responses against these very, very small protein modifications called haptens. And this is actually sort of what my project is based on. So if you take a carrier protein, let's go back to RSA, so rabbit serum albumin, and you make a small hapten modification. So NP is a really good example of a small modification. And then you vaccinate with that into a black 6 mouse. You'll get two kinds of responses. You'll get the response against the carrier protein, so RSA. You'll also get a response against the small chemical modification. And the reason that that's important is because now, when you're looking at the total antibody from the serum, we've talked about ELISAs a little bit, but it's basically a way to measure what your antibodies are specific for. You could imagine you could test for antibodies specific to just the protein, and you can test for antibodies specific to your small chemical modification, which means that what you can do is you can start to look at B cells that are only responding to this one particular epitope on the protein. Does that make sense? Did I explain that well enough?
I think so. Yeah. So maybe let's back up a second. Say like what we're measuring with this ELISA is we're measuring. You basically put, in this case, RSA into a well. You coat a well with RSA. Then you put in the serum from your mouse that you immunized. And then you can detect mouse antibodies that are bound to the RSA.
Right.
Versus you can compare that to a well where you've only coated it with the section that has the hapten stuck to an RSA peptide. Or does it even have to be an RSA peptide? Because you're looking at the hapten-specific antibody.
Yeah, what you'd do is you'd actually put the hapten on a totally irrelevant protein that you don't have an antibody response against.
Right. Okay, yep. So now we're detecting some smaller number of antibodies, presumably, or maybe the same number or higher. But
sure,
you're detecting what antibodies. Now, the downside of this approach, and this sort of gets back to what we were saying before about the difficulty in finding the B cell that produced that antibody, we can take the proteins or the antibody proteins that are specific for that hapten or are specific for that RSA, but then going back into the mouse and finding which B cell made that antibody, that's much harder because now we have to do protein sequencing, right? Or do single cell B cell cloning to figure out which one is producing the antibody so that we can sequence that. It's very, very difficult to sequence proteins.
Yeah.
And we're only looking at this very specific little epitope binding portion.
Exactly. Yeah. And so for basically, I guess where I was going with all of this is that for technical reasons, people studied vaccine outcomes using responses against these small hapten chemical modifications. It allowed for a series of technological advances that allowed us to push past the really general, I vaccinate and then it worked or it doesn't work, right? It allows you to actually assess some of the B cells that are producing those antibodies for reasons that we don't have to get into. But the problem with that approach is that, like I said, RSA, when you get a response against RSA, that's a response that presumably had couple hundred precursors, perhaps, that could all recognize that RSA molecule because RSA is a large protein, and so you've got lots of different interactions. And then over the course of the immune response, that's going to sort of be culled down to a select few, and that's what this paper will talk about today. When you're dealing with a hapten, it's an extraordinarily small chemical modification, which means that the number of B cell precursors that you're going to use or that are going to go into that sort of response are going to be necessarily limited because there's just not a lot of diversity there. The problem with that is that when all of these studies were done, people did want to ask the question, okay, well, we know that these germinal centers, we know we get collections of B cell precursors and they all see these proteins and they start interacting with T cells and you get a bunch of rounds of division and you get culling and you get promotion of B cells and somatic hypermutation, a lot of the things that we've talked about before. And in the end, we want to know how many B cells ended up seeding the response that we see, right? So sort of retrospectively, how many B cells did it take? How many precursors were actually used in the end antibody response? And because they used haptens, the answer was almost always very, very few. The assumption was that when you vaccinate against a protein, a very small number of precursors go into the germinal center. Those precursors recognize pretty well. They undergo a lot of somatic hypermutation. The mutants that no longer bound were called off. And in the end, the assumption was that a germinal center is going to almost always be entirely derived from a single B cell precursor.
And maybe we should say what a germinal center is. I know we've mentioned them in a lot, but basically this is in a lymph node. When a B cell gets activated, that B cell will proliferate. So like germ, the sort of root word, is about proliferation, it's about replication. And these germinal centers are things you can physically see. If you look under a microscope at a lymph node with some pretty rudimentary staining, you can actually see these dense pockets of cells that are rapidly dividing. That's been known about for a long time. And so that's the germinal center, where we have B cells, but also some accessory cells that are like providing the stimulation signals, some DCs, some T cells. And so that structure that you can sort of see the gross morphology of, even without specialized staining, that's called the germinal center. And so what you're saying is that it's been believed that a germinal center, once it really gets ramped up, that germinal center is derived from a single parent B cell.
It almost becomes monoclonal. So the assumption was that, and the way that I like to equate germinal centers when I first describe them to people is it's a process of natural selection. It's probably the best example of natural selection I can think of that happens over the short term in the human body, because basically what you get is you have a limited number of survival signals to go around. You have a bunch of different B cell precursors that are all sort of specific for the thing that they're responding against. And there's a survival of the fittest. The B cells that are best activated swallow up all of the survival signals, and the B cells that aren't well activated just die because of a lack of survival signals, and you just get this feed-forward loop where the best ones keep mutating and keep mutating and keep mutating, And in the end, the assumption was that the winners, quote unquote, of that germinal center were almost always going to be derived from a single B cell precursor that was then deemed sort of the winner of the germinal center. And so, like I said, for a long time, this carrier hapten approach was the standard. It was basically technologically the pinnacle of what you could do. And I actually work with somebody at Emory who did a lot of this work, Joshi Jacobs' lab, who I think at the time was a postdoc, did some really, really good work on this in the early 90s. But now we're in an age of mouse genetics, and we've got some really cool imaging technology that Gabriel Victora is fantastic at implementing towards immunological questions. And the result is this paper, which essentially turns it on its head. And I'm going to not bury the lead here and basically say that what this paper ultimately finds, and we'll go through the figures, is that mature germinal centers may or may not be monoclonal in nature in the end. So there are germinal centers where if you look, all of the B cells will have been derived from a single precursor. But in the case of complex protein vaccination, more often than not, there is heterogeneity in the germinal center. And that actually has profound impact on the way that we see vaccine biology, because if you think about the implications of a monoclonal germinal center for every protein that you inject, it means that that precursor was only targeting one part of the protein, right? So when that protein entered into the germinal center, that B cell precursor picked out one small part of that protein and said, I am specific for this. And then it divided. And then it said, oh yeah, that was a good division. Now I'm really specific for this, right? And it kept doing it and kept doing it and kept doing it. And so you get these mutations. But it's unlikely that those mutations are going to ever truly change the position of the B cell's attachment to that protein. It's more likely that you're just increasing the affinity of the B cell for that part of the protein.
Right. And most of the mutations, we should say, are going to decrease the affinity.
Sure, and they'll all die.
But in that natural selection process that Matt described earlier, most of those mutations that are going to lose affinity, they're going to also lose their survival signals, and they're going to get culled.
Right.
So you are getting increased affinity over time through selecting for the positive mutations, but the chance that a single mutation is going to allow you to bind to a completely different part of the exact same protein is really
unlikely.
rare. And I should say, I think we forgot to say the title of this paper, and you were talking about the things that go into a good science paper, and one of them is also a title that doesn't actually explain the important part of what the paper is doing. And this paper is true to form. So the title of the paper is Visualizing Antibody Affinity Maturation in Germinal Centers.
Right.
Which is true.
It is true. But
it's not really the cool part.
He's not one to overstate his science. I actually really appreciate that about him. So basically the ending implications of this finding that these end germinal centers are not going to be monoclonal helps explain really why we see these sort of polyclonal targeting, the polyclonal targeting of injected vaccinated proteins, right? So it starts to explain how we're getting diversity of targeting rather than a single response to a single part of every protein that we inject. So with that, I think we should probably jump into it here.
Yeah.
So if you're actually following along with this paper, the first figure is going to be totally overwhelming. And it's overwhelming because of the vast number of colors that are used. And so Kevin mentioned that there's some cool application of, it was neurology, right? That this was first introduced in. And so basically, in order to trace how the brain developed in mammals, some researchers, I can't actually remember where they were from, I should know this, but some researchers essentially created a mouse that they would use for lineage tracing of cells.
So it turns out that Jeff Lichtman, who is the lab is the person in whose lab the brainbow is developed. He's here at Harvard, and so So the brainbow takes advantage of a couple of really cool genetics techniques, and it cobbles them together. So one of them is this diversity of fluorescent proteins that I don't know if we've gone into a great deal of detail, but in biology, people have probably heard of the green fluorescent protein. So this is a protein that was isolated from jellyfish that is fluorescent green, and people have made huge numbers of modifications and also found other examples of similar genes in other organisms. And basically we have this array of different fluorescent protein colors that we can express in cells, and that allow us to see with some advanced microscopy and lasers and filters, allows us to distinguish between different colors. Okay, so that's one thing. Another thing is this system called Cre-Lox. And so what this is Cre is something called a recombinase that is a protein that basically cuts out chunks of DNA, and recombinases have been found in all kinds of different places, but basically they can, the Cre system will recognize DNA that is flanked by elements called LOXP elements, and it will either cut out or invert the DNA that is between the LOXP sites. And people have used this to their advantage by basically like surrounding a gene with LOXP sites and then expressing Cre under promoters that are only expressed in certain cell types. So you can, for instance, have Cre only expressed in T cells if you put it under a promoter for CD3, which is only made in T cells. And then any cell that expresses CD3 will also express Cre. The Cre will go find whatever's flanked by the LOXP sites and cut it out. And people have used this in all kinds of awesome combinations to make like reporter mice and to make cell-specific knockouts of things. And it's a really cool system. And so the way the brainbow works is sort of using multiple genes for different fluorescent proteins or combinations of fluorescent proteins, and using some clever genetics that we're not going to go into because frankly I don't fully understand all of it, basically allows you to get Cre expression in a particular cell type. The original paper was in neurons. This paper is using them in B cells.
Right.
And essentially it's random to the point where each cell, to the first approximation, is going to have a slightly different fluorescent color because it has a slightly different combination of fluorescent protein expression.
Well, I think there's actually 10. I think that there are 10 discrete combinations, right? So basically, or there are 10 discrete identifiable combinations. So basically,
right. So
for
we're
each
limited by our filters and lasers and how much we can discriminate, and also by it's using a certain set of fluorescent proteins that have different overlap.
Yeah, it's 4 fluorescent proteins, and then you obviously have 2 alleles, right?
Mm-hmm.
So on allele 1, when Cre gets turned on, allele 1, you choose one of 4 fluorescent proteins, and in allele 2, you choose one of 4 fluorescent proteins. And so those two choices in combination with each other sort of give each individual cell its own distinct signature.
Mm-hmm.
And the beauty of that Cre recombination system is that is a germline modification, right? So not only does the cell that expresses the Cre take on that color, but all of its eventual progeny also will take on that exact same color. So if I
don't know if we should say germline because it's only in B cells.
Sure, it's
germline from the perspective of the B. It's a DNA level.
Yes,
so
it's a
anytime
DNA
a cell replicates, its progeny are all going to have the same one.
Exactly.
Right.
So you can imagine that if a single B cell seeds a germinal center in a way that we were talking about before, if one B cell walks into a germinal center and then 6 weeks later, all of the B cells that are now producing antibody in that germinal center came from that first B cell, then the entire germinal center will be one color because that initial B cell was one color and all of its offspring are one color. And that is... Oh, go ahead.
I was going to say, so we should maybe say that if you see a germinal center that is all one color, you can't necessarily exclude the possibility that there were 2 or 3 B cells that seeded it that all had the same combination, because we have tens of thousands of B cells and only 10 colors. So there are going to be some overlaps.
Of course.
But if we see different colors in a germinal center, then we can know.
Right. And they also do some really cool stuff at the end here to start to look at that possibility as well.
Yeah.
So basically, in this first figure, they're just sort of showing proof of principle. And I think we've actually done a pretty good job of describing essentially what they're going to be doing here. They're going to be vaccinating mice with complex proteins, and they're going to be looking at B cells, and they're going to ask the question, when you have a mature germinal center, are those B cells from a limited number of B cell precursors, or does it look like your mature germinal centers still have a lot of heterogeneity in reference to the number of precursors that they started with? So in A, they're really just looking at this these confetti mice is what they're terming them, and so you can see all of the nice confetti colorings of all the B cells. And then they do a quick immunization of the popliteal lymph node, and what they find is that some areas of the popliteal lymph node are, surprise, surprise, one color, and they'll get back to that later. But even in unimmunized lymph nodes in the mesenteric lining, and so mesenteric lymph nodes are famous for sort of having all of these ongoing germinal center responses, even if you haven't been infected with anything. I mean, presumably they're infected with something, but these are often subclinical infections. So they look, and just at steady state, these mesenteric lymph nodes also have these clusters of single cellularity. And so I guess as a proof of concept, it probably was pretty exciting to see that you were getting these zones of single colorness. So then
you gotta imagine, like, first day you like sat down at the microscope and stared at this lymph node. Like, it's so obvious. There's just a big friggin' pink spot in the center of the lymph node. Like, that's amazing.
Yeah, no, it's really cool. And we used to do joint lab meetings with these guys, and they would talk about some of these experiments, and it was mind blowing. It's really, really cool stuff. And I think the limitation on this paper was largely breeding. Like, some of these mice are really complicated genetic awfulness. And I think they spent a lot of time trying to get these mice together for this paper. They have one at one point. Did you see the B18 confetti mouse? Nah, we'll get to there eventually. But anyway, so basically what they show is that they get, as expected, they get a good diversity of B cell colors in the germinal center, or in the lymph node in general, but sometimes in these germinal cells, or in these germinal centers, you can get single colors, essentially. And they find that if you go ahead and you vaccinate with, I believe in here, it's either OVA or CGG. I think it's chicken gamma globulin. Globulin, excuse me. So they look at skin draining lymph nodes 15 days after vaccination with chicken gamma globulin. And what they're showing here basically is 3 versions of germinal centers. One shows almost no heterogeneity in the middle, right? So it's almost entirely purple. On the left, it shows some. There's a lot of orange, but there's also a lot of other cells at day 15. And then on the far right, there's essentially no homogeneity at all. So they're completely heterogeneous. It's almost impossible to distinguish the germinal center from the background.
Yeah, I was going to actually ask that. How did they distinguish the germinal center here?
So basically the control that they're using here is they're staining for 2 things. The first is they've stained for the protein CD35. And so CD35 is present on follicular dendritic cells, and CD35 staining gets really, really bright when you've got germinal center staining. And so you can often use CD35 on the follicular dendritic cells to know that there's an active germinal center going on. The second thing that they use in this case is more of a negative stain, which is always tricky with imaging, but sometimes you just have to do it. So it's known that when you get an active germinal center response, the only cells that are going to be in the germinal center are going to be B cells that are participating in the reaction. So this is called exclusion, basically. So any B cells that are not involved in the germinal center are going to be excluded from the germinal center. And so what they're showing here is they've co-transferred irrelevant B cells that aren't going to be participating in the germinal center reaction. And so those B cells are excluded from the germinal center site. And what they're showing there is that, sure enough, there is CD35 staining, and there are not these co-transferred B cells.
Okay.
So yeah, a bit tricky, but I buy it. I feel like I think they've done a pretty good job of assessing here. So that's, I mean, basically figure one, proof of principle. You can find germinal centers that both have one color and also have lots of colors. So the question is,
and
is
they
it
also
true?
quantify this in part E. So they show us representatives of the high heterogeneity, the low heterogeneity, and the like single colony. But you can then also see they have quantification of it. Looks like quite a few mice.
Yeah.
And you can basically see a range from where they say like they assign one color the dominant color, and they say what percentage of the staining in that germinal center is the dominant color, and it goes from looks like nearly 95%, which even if you knew it was a single B cell, because this is a three-dimensional thing and you imagine there's probably some, like I wouldn't expect much more than 95%, even if I knew it was from a single B cell.
Right.
And then it goes all the way down to like only the dominant color only representing 20%
Right.
of the overall germinal center.
Right. So yeah, you basically got an entire spread.
Which I guess if there's only 10 colors, if it was totally random, you would expect 10%.
Yeah, sure.
Of the dominant color.
Sure. Yeah, you're certainly going to get some clones. And the reason, so I think going back to why even just this figure is important. If you go from the original understanding of how germinal centers are supposed to work, if you go into the old literature from the late 80s, early 90s, and you made this mouse and you did the experiment, what you would expect is that 100% of these germinal centers would have one, maybe two colors in them.
Right.
And the fact that they're finding these fully mature germinal centers with a huge amount of heterogeneity still is a major shift from where people would have expected.
Mm-hmm.
So basically, this suggests that these germinal centers are coming from a diverse set of B-cell precursors, but you have to answer that sort of directly. And for this, we introduce another cool piece of imaging technology, which is photoactivatable GFP. So now basically what we're going to do is we're saying, okay, well, we see that at, I think this is day 15? No, day 6. So their initial data suggested to them that some of these germinal centers are coming from a very diverse set of precursor B cells, right? So if you take an early germinal center, you'd expect that if you sorted all of these B cells, you'd find a huge diversity in their B cell receptors, right? And so that is what they did. They're going to get into some deep sequencing here, but basically to sum it up, essentially what they did was they took B cells that were participating in germinal center reactions, and they went in to a live mouse, found the germinal centers. So these are reporters for only activated B cells. So all of your activated B cells are going to be red, okay? So all of your activated B cells in a single germinal center are red. You go into the mouse and you do live imaging on that mouse. So now you're looking into the organ as the animal is alive, and you find a germinal center filled with these red cells. Then you hit just that germinal center with a laser
with
lasers. You hit that germinal center with lasers at the right frequency that allows them to turn on a second fluorescent protein, GFP. So we call this photoactivation of GFP. This is important because if you're studying B-cell precursor diversity and you take an entire lymph node and you just say, I want to see the precursor diversity in B-cells in this lymph node, you have no guarantee that germinal center 1 is going to have a similar B cell to germinal center 2, right? So you have no way of knowing what the actual diversity is within any individual germinal center, which means you need to only get B cells from one germinal center. So what they do is they'll go in, they'll take these, they'll find an activated B cell cluster using the red fluorescent protein. Then they will turn on the green fluorescent protein only in that cluster of cells. Then they pull out the lymph node and sort on double positive B cells. So these are activated cells by the red, and they're activated cells that were in the single germinal center by the green. And then they just sequence everything, right? So they basically go through and say, okay, well, what did the B cell receptors look like?
Right, so this is like, I'm just going to combine like 4 really awesome technologies together and show some badass data.
Right. And yeah, without getting too far, I feel like we've tried to explain really difficult sequencing, like deep sequencing data before. It never goes well.
Yeah.
So
I have
suffice
a
it to
couple of things on
say,
my blog that I wrote at one point about microbial diversity, and this is sort of, I can just point people to that. So
yeah. So we'll do that. But
RNA-seq is rough.
yeah, it's a little tricky, but basically what they find is that even within an individual germinal center at day 6, there is a huge diversity in the types of B cell receptors that are participating. So it's not that a couple B cells are going to wander into a germinal center and just start this response, and that the end of the germinal center is sort of reflective of the input. What's really happening is you're getting a huge number of different B cells that walk into the germinal center, and over the course of that germinal center response, it is going to be restricted, but it's going to be restricted from a very large pool.
Yeah, can you explain a little bit about how these pie charts are put together? Because it was a little bit confusing at first when I looked at it. It looked like every germinal center has like 30 different clones, but I think that they're clustering stuff that is like things that derive from the same parent through somatic hypermutation in some way, but I don't quite understand how that's happening.
I think, if I remember correctly, I think each slice does represent one clone. I'm just trying to
Colored
remember
slices represent clones that were found in both germinal centers, upper and lower, from the same popliteal lymph node.
Oh, okay. So yeah, so you've got so they've pulled 2 germinal centers basically from each of the popliteal lymph nodes, right? And so what they're showing is the different clones that make up both of those germinal centers. And so anything that's colored is an overlapping clone,
Right.
which means that germinal center A is actually contributing a B cell to germinal center B that is then participating in that germinal center.
Right.
Reaction.
Was that known before, that you have this cross seeding of germinal centers?
If it was known,
like,
it's extraordinarily recently known, but I don't know.
Like B cells are migrating between germinal centers. That seems contrary to what I feel like I learned.
Yeah.
Not that I studied this in any great detail, but I was like, my naive assumption about a germinal center is that you have a dendritic cell, it's got some antigen, wanders into a lymph node, you find the one B cell that detects the antigen, and that B cell sort of proliferates and forms the germinal center.
Yeah,
like,
and I think
then it's like contained.
Oh, go ahead.
But it seems here like not only are there multiple B cells interrogating the DC with the antigen, or are the DCs, the FDCs with the antigen. Not only that, but you've also got B cells that are like getting activated in one germinal center and potentially migrating over and finding a different germinal center and continuing to get activated.
Yeah. Yeah, I mean, I don't know. I know that this was a topic of conversation when I spoke with Gabriel about this a while back, but that was a while back. And so I assume that if this is not the earliest formal proof of that, then it is one of the earliest, and I suspect that it is the first, but I could be wrong. So anyway, it is an interesting, and I totally agree with you, this does not line up with my understanding of the way that I learned it either. So you are getting sharing of B cells between different germinal centers, but what's interesting to me is actually the lack of overlap. So you've got how many different slices here? Popliteal lymph node 1. So what, 20? 30?
Yeah, it looks
20,
like it.
30. And then in germinal center 2, probably 40.
Do they not have numbers? What are those numbers in the parentheses in the center?
That's a good point. 27.
I don't know what the 27 out of 40 means. It's probably in the figure legend. We should know this. Numbers in the center of each chart are the number of clones observed divided by the total number of cells sequenced.
Right. So
they only sequenced 40 cells that they pulled out, which may seem like a small number, but even there you have basically 27 different clones when you only sampled 40.
Right.
So who knows, if you sampled 100, maybe you would get 50 different clones, but it's hard to know.
So back of the envelope calculations here, right? So how many germinal centers do you have per lymph node? Probably 6? I would say
Sure.
6 to 10, between 5 and 10, somewhere in there. And so if they sequenced 40 cells, got 27 unique hits, obviously as you sequence more and more and more, your unique hits are going to go down.
Right.
But you're not seeing a ton of overlap between germinal center 1 and germinal center 2 in these two popliteal nodes. I mean, this puts the number of potential B cell precursors in the hundreds.
Right.
Right. And that is a big number. That's a really big number, because if you think about
conservatively,
right,
hundreds.
Right. Exactly. And so you're getting a ton of diversity at the front end of these things. And one of the things that I like to think about in my own project is you can't possibly have that number of T cells that are also specific to this protein, right? It means that the B cell diversity in response to this protein is going to be much higher than the T cell diversity. And it does interest me how those two things interact. So
do we know that for sure? We know that the number of antigen-specific T cells is typically lower than the number of antigen-specific B cells for a given protein antigen?
So assuming the same amount of diversity in B cells and T cells. So chicken gamma globulin, I think it's about 60 kD, somewhere in there.
Okay.
Well, okay, let me go back to my model system. RSA? RSA is about 65 kD. Okay, it's about 600 amino acids in length, right? So if you look at that 600 amino acids in length and you look at T cell help, right? So you're looking at MHC loading. You've got 600 amino acids and you're looking to load 14 mers into MHC2 binding domains.
Right.
Of those 600 amino acids, so obviously you need a 14-mer at the tail, so you're down to about 586 amino acids. Only 1 out of every 20 or so amino acid stretches are going to actually fit in the MHC, and a very small proportion of those are going to fit well. And so the way that people discuss it, the assumption is that there are probably between 2 and 10 dominant T cell clones that will actually turn into the fully functioning immune response.
Okay.
If that's the case, we're talking about orders of magnitude in difference between the B cells and the T cells. And so one of the interesting things that I look at now is how that potential restriction and how that bottleneck in T cell help might play out into the B cell response.
How much of a typical germinal center is T cells? Is T helper cells?
So you're talking. I don't have a good answer.
Are T cells being activated also in germinal centers?
There is co-activation, yeah. So TFHs, T follicular helper cells, are going to be dependent on B cells for stimulation. I guess not totally dependent, but yes, they will be activated by their B cells as well.
I guess what I'm asking more directly is, could you functionally do these same experiments given the comparable mice for these things in T cells? Could you also find a zone in a lymph node that has a cluster of T cells?
I think you certainly could, and I think someone should do it, because I think it would also be an extremely interesting contribution to the field. Yep.
Get on it, Gabriel.
Yeah, seriously. All right, so now we've basically shown that in these early germinal centers, 6 days after immunization with chicken gamma globulin, you don't have just a couple precursors. You potentially have hundreds of B cell precursors that are going to seed the starts of these germinal centers. And so then the obvious question is, okay, well, you start with hundreds. Where does that end you, right? So the next figure deals with basically exactly that. So now we're going to take advantage of another genetic manipulation tool called tamoxifen induction. So basically, we've been looking at these confetti mice, and they've been using confetti mice that the genes are turned on just in general cells, and then you've got B cells only that you picked out. Sorry, in figure 2.
In figure 2, they're not using the confetti mice. They're using
it's just straight. So figure 1 is essentially just MX1 confetti.
MX is interferon stimulated. So I think what they're doing is they have Cree expression downstream of this. This is what we were talking about before, where like you can basically use the MX Cree to do the Cree induction ex vivo, and then you transfer in cells, because this is B cell specific, right?
Yeah, I'm trying to get to the actual. So these are Rosa 26. Yeah, so these are Rosa confettis originally.
Yeah, but the Rosa 26 promoter is just a
right. It's a general
ubiquitous promoter.
Right, exactly. So this is where they get specification into the actual B cell promoters.
Right.
Right. And so I'm just trying to figure out what the there we go. AICDA Cree ER2. 26. Oh, that's so God. So they've got the estrogen receptor, right? Is that what it's going on? So they've got the B cell activation gene. That's the AICDA gene.
Mm-hmm.
And on that, they've got...
Well, the estrogen receptor is what responds to tamoxifen.
Right, exactly. So they've got the Rosa confetti, but that's floxed? God, this is complicated.
I'm definitely including this audio just because it's nice for people to see that sometimes we don't know what the hell we're talking about sometimes.
Yeah, I mean, so basically the end result of this is that you're going to have B cells that when they are undergoing a normal germinal center response, the Cree is not yet induced. So then what they can do is they can introduce tamoxifen into the mouse at a time of their choosing, which sort of turns on the Cree. So they'll vaccinate these mice with chicken gamma globulin, and then 5 days into the vaccination, when they know the germinal centers have already started forming, that's when they induce the tamoxifen. So basically, it's sort of like an old pulse chase experiment where you pulse on day 5 with tamoxifen, all of the B cells recombine their fluorescent proteins, and then you follow from day 5 to the end of the germinal center. And so you're looking to see how the clonality was basically culled in these germinal centers. So you say, I have X diversity at day 5. What diversity do I have at day whatever, 6 weeks out?
Right, because it should be noted that at this day 5, what we're going to have is we're going to have a B cell that had 2 B cells that both came from the same parent B cell, the same initial VDJ recombination. In this system, those 2 B cells, even though they came from the same clone, could have different colors in this system because we're inducing this color recombination after a huge amount of clonal expansion and hypermutation has already occurred, right? As opposed to in the first one where every single VDJ recombination in principle would have the same color. Here, what we're saying is the same two B cells that had the same parent could have different colors, and so now we can follow individual B cells from day 5 on.
Right, exactly. You're just basically starting the whole process at day 5. Right. So you haven't undergone a huge amount of division yet. So there shouldn't be. So when they did the diversity experiment in Figure 2, mm-hmm. right, they were doing that at 6 days after immunization. Right. So they basically already shown that at 6 days post immunization, there is still a huge amount of clonal diversity. Right. So what they do is they induce tamoxifen at day 5, which is a day earlier than that. So these B cells are still from a very diverse background. Right. And what they want to know is from the background at day 5, how many of these germinal centers will progress into sort of these monoclonal, everybody came from the same precursor germinal center. Right. And so, they do this in a fairly straightforward way. They essentially just create an index of diversity, right? And they do this, and basically what they do is they go through whole mount lymph nodes. So these are lymph nodes that have been cleared and imaged, and they capture the entirety of the germinal center, and they essentially count cell by cell in some cases, and they categorize how many germinal centers there are, what sorts of colors make up each germinal center. And as a control, this is really interesting, as a control, they have the same mouse, this confetti mouse, except on a B18 background. And a B18 background basically means that all of the B cell receptors in these mice are the same. It's a knock-in receptor, basically. And so if
is that a
you
CGG specific B cell?
It is not. They vaccinate these animals with NP ova. It's actually specific for the hapten NP that we talked about before. And so you would expect that because all of these B cells came from a limited number of precursors, you'd have more monoclonality, just like we were talking about as far as our assumptions leading into this paper. If you have very few precursors on the way in, you're not going to expect many precursors on the way out. And so basically what they do is they measure the amount of germinal center color diversity in each individual germinal center against a set of germinal centers in these B18 background mice.
Yeah,
and
so I'm a little bit confused about that statement, because what you would expect where every single B cell receptor is the same, because you're inducing the confetti at a later time point, what you would expect, because the confetti, the color expression has nothing to do with the B cell receptor gene. So even though all of these mice have the exact same B cell receptor,
right,
less a little bit of, a somatic hypermutation that's going to go on, you should expect to see basically what we were talking about before, where like totally random, you would expect 10%.
Sure.
You'd expect so they have this thing where they're looking at diversity, where basically 0.1 means you have all colors expressed equally, all 10 colors expressed equally, and 1 means that you have a single color that is completely dominant.
Right.
So you'd expect in the if there is absolutely zero selection and it's all random in the what's the mouse, the B1? B18. You would expect it to stay at 0.1 the whole time.
Would you? So you're still going to undergo somatic hypermutation, which means that some of these receptors are going to become slightly higher affinity than others. And so what they show is, yes, you do get diversion away from that 0.1, but it's pretty standardized in that you don't get these huge outliers where a single B cell takes over the whole germinal center. Right. So I think because of somatic hypermutation, you do get some competition in these germinal centers, but not the same sorts of runaway competition that you see in the diverse precursor set.
Yeah, and I almost think that, so this B18 B cell receptor presumably came from a monoclonal, from a B cell that was single cell cloned that had been vaccinated against the hapten.
Yes.
So it's got a B cell receptor that has already undergone somatic hypermutation in one mouse.
Yeah.
And we're basically starting the B cells off at that point.
Yeah.
So I guess I would expect that if we expected that that was the best B cell you could possibly make, which it probably isn't, but like let's just assume naively that that's the case, then you would expect to basically stay at 10%. And what you see is you see some mice where it does stay at 10%, so you do have basically randomness, but you do see in some lymph nodes that you get an accumulation, and I actually suspect that a huge part of that is just essentially random noise.
And it could very well be. Could very well be.
But
you definitely...
The key is that in the non-control mice, so in the ones that have this diverse response, you see a lot of mice that basically accumulate, they basically start to become dominant, even starting at day 5. They get better and better, and so you have like these runaway hits that are just like, they clearly started with some, like at some point where they were poised to become super high affinity.
Right.
Or I guess that none of the other B cells started at a place where they could become high affinity.
Exactly. Yeah, you actually have really no idea as to
Yeah.
why any individual B cell won, but
Right.
you do get some situations by hook or by crook where some B cells just go in and just kill it.
At least relatively. We don't know if they actually killed it, but they are relatively dominant. Because it turns out this is one of my favorite things talking about evolution and natural selection is that, like, particularly in a case like this, you don't actually need to be the fastest elk. You just need to be faster than the slowest elk.
Yep.
And that's true in B cell responses as well. If you have one B cell, even if it sucks, if all of the other B cells suck more, that B cell is going to get all the survival signals.
What is it? In the land of the blind?
The one-eyed man is king.
Yeah.
My other favorite is the if the zombies chase us, I'm tripping you.
Right. Yes. Yep. So
we're
don't
moving far
be
afield
out
now
with Kevin during the apocalypse. But what actually interests me about these figures, and I think we've hit on it a couple times, so I don't want to belabor the point too much, but I'm actually surprised that more of these germinal centers are not monoclonal. The surprising part of this data, and I think this is what you were getting at, where you're saying, well, I'd expect a little more diversity in the B18s. I am super surprised that there is not less diversity in these normal precursor responses. Like the percentage of the time that there's like this runaway B cell that just dominates a germinal center is surprisingly low in what I would expect going into this paper.
Well, but the thing is
what
you
makes
have,
this data set cool.
At day 5, you've got, like, from the original figure 1,
Sure.
you might have a germinal center. I actually think the ones where you see less diversity in this experiment, so in other words, you see numbers closer to 0.1, I think what that tells you is that by day 5, you were a super low diversity germinal center.
So
does that make sense? Like you came from a single clone, huh?
Do you see that though in figure 2? Because so this is a day later in figure 2 where they're assessing germinal center diversity.
Oh, that's
true.
And
there's not a single germinal center in that set that has fewer than 20 precursors.
That's true.
Yeah. So
that's a fair point. I was thinking
totally
about the first
saying,
set, but yeah, the sequencing is definitely better evidence.
So then they get into the last piece of awesome technology that I want to talk about, which is laser dissection.
Yeah.
So it turns out that a thing that you can do is you can take a live lymph node and you can cut it up with lasers. They're doing imaging under a scope. Right. So you've got a system where you can see these germinal centers, and you want to ask the question very similar to our GFP inducible system before. You want to see what the clonal diversity of each individual germinal center is. Right. So you go into this whole mount where you can visualize each individual germinal center, and then basically what you do is you just take a laser and you cut out each germinal center. And then you sequence everything, all of it. You do single cell sequencing, IG sequencing, for basically everything that was in that germinal center. And in doing that, you can construct these gorgeous maps of how B cells came from their precursors, right? You can basically construct a phylogenetic tree or a series of phylogenetic trees where you can say, look, we had, these 40 B cells that we sequenced. We know that these 20 are related to each other. Here's the putative precursor that they came from, whereas these other 5 are from a different potential precursor. We know that, and they would have been this color based on the genes that we see. They basically go in and sequence the crap out of these things and show exactly how the lineages of each germinal center related to their precursor. And that is so cool.
Yeah.
It's so cool to me.
By the way, are they, not to take away from this at all, because it's super friggin' cool, no doubt. It occurred to me that maybe they're not doing RNA-seq. In fact, I think they're not doing RNA-seq. I think that they are single cell.
They're doing IG sequencing, I think.
Cloning. They're sorting single cells into wells.
Yep.
And then they are doing PCR on the variable region.
Yep.
And then just doing Sanger sequencing on that.
Yeah. Yeah, I think you're probably right.
Because otherwise, the data would look very different. They wouldn't only be sequencing subsets.
Yeah. Yep. I think you are right. I think you're right. So basically, now you've got these two metrics, right? You know what color each cell was, and you know what the variable region of their IG looks like. And from that, you can construct these beautiful maps following progeny, essentially, in the germinal center. And if this is not definitive proof of something like this, then I don't know what is. This is amongst the most definitive experiments I think
this is inarguable.
Like it's beautiful. It's absolutely beautiful. So everything that we've said to this point sort of holds true to this figure. You can find germinal centers where there's a lot of color diversity at the end. I think that this is 20 days following. Make sure that that's right. 10 days after tamoxifen, so that would be 15 days after immunization. So this is 15 days out in a fully formed germinal center, and you can find germinal centers that are all the same color. And then if you go into their actual sequences, you can see that all of those same color B cells were related to each other at the beginning. So there are germinal centers that basically, in the end, come from a single precursor. But you can also find germinal centers that are highly diverse in their coloring, and then if you go back into their actual variable region sequences, you can show that they came from different precursors.
Yeah.
So to me, this is sort of the critical figure. I think that if you just had this figure in front of you, you could pretty definitively give the main thesis of this paper. I would say that they go on in figure 5. You might... So they go on to figure 5 to test whether this issue of diversity from the precursor has a functional implication on the potential affinity of each individual clone. So one of the thoughts has always been that the reason that you need this sort of monoclonal end response is that you need a B cell to go through X numbers of rounds of somatic hypermutation before you can get a high affinity B cell. And it's just the high affinity B cells that are the good ones, quote unquote, for protection. And so you might imagine if you have a diverse germinal center from lots of different precursors, maybe that diversity has sacrificed somehow the affinity that any one B cell will have for its target. And they basically show that that's not the case at all. So what they show is essentially, yes, you do get lots of affinity maturation in germinal centers that have pretty much a single clone, but the affinity of that clone for its target is not so different from the affinity of all the clones from the diverse germinal centers.
Right.
And that actually maybe speaks to your question before. Maybe all the other B cells just suck in the monoclonal germinal centers. And so when you don't have a lot of good B cells around, you need a ton of affinity or somatic hypermutation in order to sort of get you to that affinity threshold.
I wonder if another possibility is that you mentioned the T cells before, and this paper doesn't talk about them at all, but like I wonder if you have a single T cell, you have a limited number of potential B cells that you can support, essentially, right? So I wonder if the more diverse germinal centers also have more diverse T cell help.
That would be super interesting to ask that question. And I know that this lab has published papers using basically tracking T follicular helper cells between germinal centers and showing that they can sort of be bounced back and forth, like in vivo tracking essentially of TFHs. And so I think assessing the diversity of T cell help and how that influences the diversity of B cell response is something that I am personally interested in. I'm sure Gabriel's interested in. Actually, I sort of hope to talk to him about some of the things that I'm doing, because this paper, like, there are so many techniques that I could use from this paper to address some of the questions that I have. So anyway, fantastic.
Super rad.
Fantastic. I really liked that one. Yep.
Yeah, it's a super cool paper and so pretty. Like, there's so many, I can't even decide which image to pull to post with the episode, because there's just Like there's always this thing where like if you're a biochemist like me, like everything you can show is just Western blot after Western blot, and like that's cool for a certain subset of people, but the overlap of the size of that bubble of people who are interested in looking at Western blots is just dwarfed by the number of people that you show a pretty blue, red, and yellow rainbow colored fluorescence micrograph. It's just like, you win.
There's a slightly different impact.
You win. Yeah.
Yep.
It's pretty amazing.
That's why we draw cartoons.
Yeah, it's true.
Yep.
Excellent. Well,
all right.
Thank you for suggesting this, Matt. This was a super cool paper. And again, like the way that this was suggested is 3 days ago, I was like, hey guys, we should record on Tuesday. And does anybody have any paper suggestions? And Matt was like, we should do this one. Okay.
Yep. Done and done.
Easy.
Yep.
So this has been Audiommunity, episode something or another. I guess we're not saying episode numbers anymore.
No.
But it was recorded on May 3rd. You can find us at emmunity.org, emmunity.org, and as we said on the last episode, all over the internet. You can subscribe to us in iTunes, Stitcher, Google Play, RSS feed from our website. You can look us up on Facebook. Kate keeps saying she's going to make a Twitter feed, but she doesn't even update the Facebook page that I made, so that's probably never going to happen.
Seems unlikely.
What else? Oh, I should say, if you like the show, please rate us on iTunes. Like, even if you listen to us on one of these other positions, the iTunes library is really what matters in terms of podcast ratings. So please go leave us a review. If you write words, it's better, but even if you just give us some stars, hopefully 5 stars, that would be great to improve our audience. Please send us feedback. You can do that through the website, or you can send us an email. The music at the beginning and the end of the show was composed by Rachel Reineck, and we'll see you next time.